All right, so welcome to today's TNT seminar. Um, for those who are new to coming to our events, TNT stands for Translational Neuroengineering Technologies and the Network. And so we're a group here at Hopkins interested in translational neuroengineering technologies, as the name suggests, and we put on seminars, journal clubs, happy hours, holiday parties, things like that. So both professional, academic, and social events for people who have this interest to come and learn and spend time with each other. A couple of things. Thank you for joining, and we have more events coming up. So on November 11th, we're going to have Paul Marasco from Cleveland Clinic speaking to us, and we'll have a journal club as well in November. So look out for those events in the near term. A couple more things during the talk today. Feel free to jump in any time and ask questions. You can just jump in over the audio or type it in the chat or raise your hand, whatever you feel comfortable with, but feel free to jump in whenever. And the last thing is that there will be a poll to get some information, and please fill that out. It will be put out near the end of the talk. And so with that, to introduce our speaker today, we have Francesco Tenore, who works at the Applied Physics Lab, working on neural interface research and development. He got his PhD here at Hopkins in the ECE department, and so we're very excited to have him talk about translational neural interfaces. He also goes by Frost, so feel free to call him that. That's what he's comfortable with. So with that, thank you, Fra, for speaking, and I'll leave it up to you now.
Thank you, Mark. Thank you, Celio, for organizing this. Everybody can hear me okay?
Yep.
All right. So I'm going to get started on this talk for today, and I want to talk about translational neural interfaces and a little bit of the journey that has gotten us to where we are. I'm going to be focused mostly on cortical neural interfaces, and these days I've been starting all my slide decks asking the artificial intelligence what it thinks that neural interfaces are. And so I put in the phrase human implanted with neural interface, and then I added cortical neural interface to see what it would come up with. And these were a couple of the images that it gave me. And I thought, that's good examples of maybe central nervous system and peripheral nervous system interfaces. So a little bit about me. As Mark mentioned, I actually graduated. I got my electrical engineering bachelor's degree from the University of Trieste, which is in the northeast of Italy, very close to the border with Slovenia. And then I came to Baltimore for my PhD in electrical engineering. And so in these three circles, you can kind of see the three places that I've lived all my life. And so, yeah, since 2008, I started working at the Johns Hopkins Applied Physics Lab, which is, for those of you who aren't familiar, it's midway between Baltimore and DC. And in fact, it grows so much that our building isn't even pictured here. We are behind this building here. This is the Space Department building. Behind here, there's a very nice new building, which is where we recently moved to at the beginning of this year. And the Space Department, obviously, being these days very famous for the successful DART mission that completed back in September last month, actually a month ago yesterday. But the reason that I originally joined APL was more for some of the research that was going on in prosthetics and neural interfaces. That was how I got involved in some of this research, and I've been developing it. I've been working on it since I started working at APL, and it's been a very exciting journey. And I wanted to take the opportunity to share it with you. but first I wanted to go over what I want to talk about, right? We're talking about neural interfaces, and neural interfaces provide direct communication pathway to the brain's electrical activity. And to this, I want to add two things. One is I want to make sure it's clear that the communication pathway is bidirectional, right? We're intent to record, but also stimulate the brain. And this is a translational activity, which means that what we're interested in is to be able to communicate with the human brain, not just animal activities or benchtop activities. We're interested in actual communication with the human brain. That's what makes this translational. And as we'll see, there's a number of steps that need to happen in order for us to be able to use the technology that we've been developing in humans. And it's been a really interesting journey, and I think it's a great opportunity to be able to share it on this meeting. So the actual origin story stems from there. It comes from Revolutionizing Prosthetics, which was a DARPA program that began in 2006. I joined APL, as I mentioned, in 2008. And the goals for it was to restore neural function after amputation or injury. So the first two phases between 2006 and 2009 were focused on developing the most advanced upper extremity prosthesis in existence. And it was intended to be modular, which meant that it could be used for any type of amputation, but it could also be used for being controlled in other domains, such as spinal cord injury. And that's how kind of it evolved between phase 1 and phase 2. And the way I want to go through this today is to focus on each one of these kind of two pathways separately, and then then take it from there. So let's start with a few questions that I think are very interesting to address. So what should a neural interface look like? This seems like a relatively standard question, or a strange question to ask, right? What does a neural interface look like? Internally, it can kind of look like whatever you want. Externally, it doesn't necessarily have to look like this, but this is what they currently look like, right? They're more similar to, on the left-hand side, something non-invasive like an electroencephalography grid, and in the middle is one of our former participants at the University of Pittsburgh, who's implanted with penetrating microelectrode arrays. You can see that both of these are wired devices, and so this doesn't make it quite amenable to be able to use these in real world environments, right? So we want to always have that in mind when we think about how we want to evolve these interfaces. Should they be invasive or non-invasive? Well, in an ideal world, we can get everything we need from non-invasive interfaces. But there may be trade-offs associated with going a non-invasive route versus an invasive route. And we're going to explore a little bit of these. Mostly I'm going to be focused on the invasive side, but towards the end, I'll also give you some recent updates on where non-invasive technologies might be going, to see if the trade-off is skewing more in one direction versus another. What would you want it to be capable of? This is another important question, right? If you are thinking about that trade-off between going invasive, so going for a brain surgery to have these devices implanted, you want this device to be capable of all kinds of things, right? And so that's kind of how we want to think about the trade-off. The more capabilities it have, the more compelling it might be to be more risky about what you have implanted, what you use as your interface. And then finally, how long would you want it to last? When I turned 30, I had laser surgery to my eyes. I was nearsighted for as long as I can remember. And as a 30th birthday present, my parents got me a PRK, photorefractive keratectomy, so that I'd be able to finally no longer use glasses. And, you know, somewhere between 10 and 20 years later, that effect has gone. So in thinking about an invasive interface, you know, is that something that you would consider if you had to undergo brain surgery? If it's not invasive, then, you know, it's a little bit moot in the sense that, you know, even if it breaks, you can replace it with a new one. So I'm sure that many of you are familiar with this plot, which shows, in this case, I'm showing some of the non-invasive methods to acquire neural activity. And they go, depending on the capabilities that you are interested in focusing on, you can either have fast temporal resolution, but low spatial resolution, or better spatial resolution, but slow temporal resolution. This has been so far the trade-off, the trade-off in choosing non-invasive versus invasive neural interfaces. On the invasive side, that's where you start to get on the bottom right quadrant of this plot, and with electrocorticography, this still requires brain surgery, but you're placing a surface electrode array, an electrode array on the surface of the brain, and you're getting better resolution electrocorticography, neural signals, neural activity, and then if you want access to actual action potentials and individual neural activity or multi-unit activity, there's been some technologies over the past 2 decades that have allowed that too. And these were demonstrated first in non-human primates, and then more recently, particularly with the Utah array, has been the go-to standard for neural interfaces, penetrating neural interfaces in human clinical trials. So I wanted to go over some of these requirements that we're looking at. Like, in order to acquire a user's intent, what is it that we need? So I'm going to argue that high spatial temporal resolution is kind of a must. We've seen that this buys you control over multiple degrees of freedom. And the more channels and the higher the resolution you have, the spatial temporal resolution you have, the higher the likelihood that you're going to be able to control many degrees of freedom. What you use them for, you know, that's going to be up to your particular application, but you need to make sure that you can guarantee some kind of functional control. This kind of started back in 2006 with the BrainGate studies. This was work done by Lee Hochberg and John Donahue at up in Boston, and this was the first human implanted with a Utah electrode array.
Okay, so here's the cyberkinetics desktop. What are you going to do first?
Apologies for the quality of the video.
Email first.
Okay.
Open the first one.
You're going to open the first one.
And it says, congratulations, you are doing a great job.
Very good.
So here you have the first person implanted with a single Utah electrode array in primary motor cortex, and they're decoding 2 degrees of freedom that allows a person to interface with a computer to perform a specific task and have, you know, some degree of control that his injury or disability prevents him from having normally. This was how this started. So the Utah electrode array is a 10 by 10 array of platinum tipped electrodes that are spatially 400 microns apart. So a total of take up a 4 by 4 millimeters, and can acquire data at up to 30 kilosamples per second, right? And so that's what gives you that capability. Longevity of use. We talked about this earlier. We'd like, for non-invasive devices, we can just replace them if need be. But for these invasive devices, we would like to make sure that they are functional for at least 5 years. And recently there was a Wired article on, this was actually 2 months ago, that showed Nathan Copeland, who is a former participant in on the Revolutionizing Prosthetics Phase 3 studies, in which he passed the old record, which was of 7 years and a little bit more than a couple months, and he's still implanted, since 2015. This is a case of an N of 1 or 2. Obviously, we'd want to see this in a lot more than that. Right now, across the country, implanted with these penetrating extra arrays, there's probably somewhere, I want to say, in the 40 people approximately who are implanted with these devices. And so they're paving the way for where we're going to be headed, but not everybody has been able to achieve these kinds of lengths of use. Wireless communication. I think this one is also important, so that you don't have tethers. You can use the device no matter where you go. And this is actually a more complex problem than what we might think, because neural activity, especially at these constraints and these requirements, the resolution of the data that we're looking for requires a lot of data to pass wirelessly. Back in 2007, in collaboration with the University of Utah and BlackRock Microsystems, we had taken an interesting stab at this, in which an application-specific integrated circuit was flip chip mounted to the back of a Utah electrode array, and the ASIC had amplification capabilities, digitization, telemetry capabilities, and so it was acquiring power and data inductively. So it was quite a bit ahead of its time. Unfortunately, for a number of reasons, this didn't quite succeed. This is actually what it looked like when it was all fully built. This was the device, external device that was placed directly over the implant to be able to collect and configure the parameters of the array. And it turns out that there were a number of problems that made it such that it was going to require a lot more development in order to get it into clinical or translational use. We did some testing back in the day. Obviously, benchtop testing showed that the connectivity was good, but you really had to place the recording module directly above the array or the coil. And then encapsulation has been also a very troubling obstacle over the course of the years, because you have to be able to guarantee that that the body will not destroy the encapsulation as you have it implanted for extended periods of time. So what we did was we tried to take it one step, so to say, easier, and the next step, to play it a little bit more conservative, was if you want to have access to multiple, more than just 100 electrodes connected to a single pedestal, then you could have a a different type of ASIC that's basically doing for the most part, is doing just amplification, digitization, and multiplexing so that you can connect more channels to a single patient pedestal and have access to more neural information that way, instead of implanting, one pedestal for every 100 electrodes that you have implanted. And this one was also ambitious, but this one was tested in benchtop testing. We did some long-term testing, accelerated testing to see how the encapsulation would react. And unfortunately, this one too, we ran into a number of issues associated with encapsulation, and so we had to take a step back. So we've been working on this dual side, which we're trying to advance the technology, but at the same time, we want to make sure that we know what the capabilities and what the requirements for testing these devices in humans look like, and what is going to be acceptable by the FDA. This is taken straight from the Neuralink website. This is Neuralink's approach to this. They have neural threads, so their business end is obviously different from the one that BlackRock has been developing. But obviously, as you can imagine, most of the rest of the configuration, the processing, the amplification, digitization, power and telemetry module looks kind of similar to what we had back in 2007 in terms of how, what's being communicated and what and how it's being powered. That shouldn't be terribly surprising, but obviously their form factor is a lot more elegant here. Where they are not, as far as I know, they are not in clinical trials yet. They're still doing preclinical testing in order to receive the regulatory approvals to begin the clinical trials. But when they do, if they do and when they do, this is the device that they've been using. What the approach that we've been taking and using Utah electrode arrays is to play it even safer, is instead of putting all the electronics on the back of the Utah array and making a risk of being attacked by the body over extended period of time, another approach is to put all the electronics subdermally, implant them subdermally, so that communication is a lot easier because you're just under the surface of the skin, and you're a lot less risk of encapsulation issues. So this is work that we're hoping to begin soon with the VIS Center in Switzerland, who's developed this module to interface with the Utah electrode arrays. And so we're excited to see where this goes. So we talked about this wireless communication. In terms of one important requirement is that it be easy to implant. And what I mean by that is that if you do implant it, if there is a requirement for surgery, then you want to have some kind of mechanism to guarantee that you're going to be able to place them in the right place, and that you are going to have access to as much neural information as possible in the area that you've implanted, right? And right now, this is a bottleneck. This is not something that has been solved. The main way that it kind of quote unquote gets solved is if you implant more electrode arrays, then you have a higher probability of getting useful information. But I know that some teams are also working on seeing what can be done to streamline this more effectively in the OR for placement. And then I also want to emphasize stimulation. So stimulation implies that when we are interfacing, when we are controlling these external devices, we are also able to perceive contact of these external devices or information associated with these internal devices that does not necessarily require exclusively on vision, right? Vision is doing a lot of work in many of these tests. If you have the ability to stimulate the brain in such a way that it becomes intuitive to perceive what is happening, then you are likely also going to be increasing the performance of the system. And finally, number of channels. What's the requirement here? Obviously, the easy answer is the more the better. This always has to be taken with the understanding that it has to fit within FDA requirements. So if you do want to implant a large number of channels, or if you have a new technology that's able to record from many channels, there has to be a pathway for how that can get approved by the FDA. So that has to always be something that we keep in mind. And so let's talk about the other side of the pathway, the neural stimulation pathway. This is something that we've been working on also. This is since probably 2013 in collaboration with the University of Chicago. We had the first demonstrations of a sensory prosthesis in which we trained a monkey to react in different ways depending on the stimulation pulses that it was perceiving. So it was implanted with floating microelectrode arrays and Utah electrode arrays in the areas 1 and 3B of primary somatosensory cortex. And we were also indenting the fingers of the monkey's hand. And we were trying to see if we could find an effective way of drawing a correlation between the indentation of the stimulation of the skin and the electrical stimulation of the arrays through which we're delivering a stimulation directly into S1. So in order for that to be effective, two things have to happen. One is you have to be able to make sure that you can control, have access to the same locations of stimulation. So if I'm stimulating a specific area of S1, that has to be perceived in some location that I'm indenting. And then the second one is I want to be able to also deliver current that is proportional to a certain amount of pressure that I'm applying to the skin, so that I can match those pressures with the electrical stimulation. And this resulted in, again, back in 2013, this was the first demonstration. Let me set it up for you. This is a finger of the modular prosthetic limb that it has sensors capable of basically, let's say, just for now, discriminating between different pressures applied by the indenter. And the monkey is on this side and is performing the task. So it's trained to saccade in one of two directions, depending on whether he felt the stimulation in the first or the second of two intervals. So this is the force output from the modular prosthetic limb. So as you can see, if it increases, it means we're applying a pressure to the modular prosthetic limb. And if he feels it, the stimulation only comes in one of two intervals. It's either in the first one or the second one. So the monkey has to wait until both intervals are completed and then has to saccade in one of the two directions, depending on where it perceived in which one of the two phases it perceived the stimulation, and it performed extraordinarily well. So that gave us confidence that it is possible to create a sensory prosthesis. The question then began to be, what was it going to take in order for us to be able to translate into human work? And so that led to some of the research on the safety of intracortical stimulation. So we had to basically create patterns of stimulation that would give us access to as many parameters as we could probe within the resources that we had available, right? So we tested these patterns of stimulation, these different patterns of stimulations in 3 different monkeys, and then at the end of the study, the animals were sacrificed, their tissues were examined, and when we looked at whether there was a way to determine the damage that was done as a result of stimulation over the course of during the histology, right? So what we did was we tested 3 different current amplitudes, 10, 20, and 100 microamps, with different duty cycles of either 1-to-1, 50% duty cycle, or 25% duty cycle, with durations of 5 seconds or 1 second. And this resulted in a couple of papers, and the data that came from this ultimately was what is currently allowing us to deliver electrical stimulation in humans. In particular, all the parameters that we tested showed that we were going to be able to deliver this safe and effective stimulation. Maybe the only caveat that we found was that the current stimulation that could be delivered should be limited to somewhere around 700, no more than 700 microamps, but we haven't found a case in which we've had to deliver that much current. So we went a little bit through some of the technologies and the capabilities that have been developed. I now wanted to provide a little bit of a framework for how these have evolved over time. And so I started by showing you Lee Hochberg's initial testing in 2006, the first person involved in the first clinical trials with Utah electrode arrays. And that was in 2006. In 2013, 7 years later, under the Revolutionizing Prosthetics program, Jen Colliger and colleagues doubled the number of electrodes that were being implanted, and they demonstrated that with that many channels, it was possible to control up to 7 degrees of freedom. Richard Anderson at Caltech and his team implanted in a different region of the brain. They moved from primary motor cortex to Brodmann area 5 and anterior intraparietal cortex areas. And then from there, shortly after that, we were able to introduce intracortical stimulation to the picture. And so Nathan Copeland, who I showed you earlier, was implanted in 2015 with the first Utah electrode arrays with sputtered iridium oxide film for stimulation. And more recently, our team at APL and Hopkins has implanted a total of 6 electrode arrays, 4 in the dominant hemisphere and 2 in the non-dominant hemisphere. And we balanced the number of platinum and recording and stimulating arrays. And so I'm going to show you some recent data from this work. So one thing that happened during the surgery is that and I mentioned this earlier, that it's not easy to find the optimal locations for implants. We did a great job in identifying appropriate locations that would map to hand and finger areas for the stimulating arrays. The motor arrays did not have quite as much success, and so we had a little bit more constraints in what we were able to control. In this study, so we used some of the AI expertise that we have with it at APL to help assist and or supplement the capabilities that the human was able to provide through the brain interface, to show that with these bilateral arrays, it was still possible to conduct an activity of daily living that we may take for granted, but that requires a lot of effort, and it can only really be achieved if you have bilateral arrays. So, show you what this looks
Cut
like.
and eat food. move right hand forward to start. Moving fork to food.
The machine provides information to the user
Select
about
food
which
location,
degrees of freedom it's
right hand,
controlling
forward, back, left, right.
at any given time.
Poke food, move right hand down. Moving knife to left of fork. Select cut point, move left hand, forward, back, left, right. Cut food, move left hand down, move right hand right or back. Retracting knife. Scoop food, roll right hand right, moving food to mouth. Align food with mouth, up, down, left, right. Eat food, move food to mouth. Moving fork away from mouth.
So yeah, apologies for the music. I think we also have a version without the music, but so we call this, a shared control approach, where we're using the capabilities that we are able to achieve with neural decoding, and supplementing that with the intelligence built into the robotic system.
Cut and eat.
On the simulation side, as I mentioned, we had a lot of good success in positioning the electrode arrays in locations that we thought were going to be of great benefit to some of the testing that we were planning. And as you can see, we have access to on the dominant electrodes, on the dominant arrays, we have access to thumb, index, middle, and ring, and we had access to the fingertips of 3 or 4 on the non-dominant hand, excuse me. And the thresholds, the simulation thresholds that we were finding were comparable, if not a little bit lower than what we had seen in our experiments with the non-human primates. So that was all very exciting. This is a video of our participant giving us a little bit of an explanation of what he's actually feeling when we deliver the stimulation.
All right, that was the right thumb. Kind of 1 through 4 all the way around both sides. That was kind of pressure. It felt like someone was squeezing my thumb. Okay, right first finger, and it was actually 1 through 8. That was more pressure. When I say pressure, I'm trying to think what we were saying before. Pressure, it feels more like somebody's pushing on my hand, like that kind of pressure. So it's more, I guess, external. One more time. Okay. It's going to be the right kind of middle finger, section 7 and 8. All right, that was the right palm side ring finger. It was kind of right in the center of 1 through 4.
So yeah, so these were some examples of what he's feeling when we're delivering the simulation, right? And this is when you really need the human in the loop for these things. Very hard to extract this information from non-human primate studies. We also did some testing to see, because of the fact we have this direct neural interface, we're stimulating directly on area 1 of S1. We did want to find out whether which kind of stimulation modality, whether natural haptic touch or artificial intracortical stimulation, would be faster. So we conducted sort of these different types of experiments to kind of get to a determination about which one would appear to be faster. And what we found was pretty interesting. So on the reaction time to sensory stimulation, so which one was perceived faster, and participants was asked to verbally report when they felt the percept. We found that we tested two different intensities of stimulation. We tried to match the stimulation delivered intracortically to the extent that was possible. And then we did the comparisons. And what we found was that in general, the mechanical stimulation, so the skin vibration, was perceived significantly more quickly than the intracortical microstimulation, which at first we thought might be a little bit surprising, because you would expect that if you have a direct communication cortical connection, you're communicating directly with cortex, that it would be perceived quicker. But that's not what we found. A possible explanation that we gave for that is that it's possible that the slower perception speed of ICMS might be due to the fact that, with ICMS, we're bypassing certain processing stages, including the cunate nucleus and the thalamus, that might be critical to the way we interpret perception, right? And that might give an advantage. We also did a temporal order judgment task. We conducted a temporal order judgment task in which vibration and ICMS were either spatially offset or co-located, so occurring on the same location, and we wanted to see which one was perceived sooner. And in this particular test, this was a little bit trickier, because when they are co-located, if they are occurring at the same time, even though we did do what we could to intensity match the ICMS stimulation, it was still a little bit being confused with the vibration. In other words, when they were delivered simultaneously, there would be frequent instances of the participants only mentioning that vibration was detected. And so what can you do? If you do have access to stimulation, then you can do a different form of testing in which you're controlling a prosthetic limb, and you're sensing information about the object that you're coming in contact with, and maybe you can make judgments about some kind of characteristics of the object that you're perceiving through the ICMS. So the sensory information, the sensors in the modular prosthetic limb are obviously acquiring different types of information depending on the object that we're presenting to the hand. One of these two objects, there's two spheres. One of them is softer, one of them is harder, and the participant knows which one is which.
Paintball.
So he refers to them by their color as opposed to their stiffness.
Black ball. Pink ball. Black ball.
So he's able to make this differentiation, which was very interesting to us. And finally, I also wanted to talk to you about some other research that is ongoing at APL. As I mentioned earlier, that dashed line that draws the difference between invasive and non-invasive abilities. is something we're working on. So since 2019, DARPA started a program called NCUBE, which stands for Next Generation Non-Surgical Neural Technologies, in which they're basically looking for new devices, novel devices, non-invasive, non-surgical devices that are capable of acquiring neural information at the same spatial temporal resolution as penetrating, as invasive interfaces are capable of, but it has to be, as I said, completely non-invasive. And so there's three modes of doing this neural recording. Vascular responses are typically slow. They rely on blood oxygen concentration. Electrical stimulation is what we've been looking at more recently. So direct neural interfaces with cortex. But there's also a possibility of using something called a fast optical signal, which you're basically looking into movements, very, very minute movements of neurons, which when they fire, they also have very, very small displacements. And if there's a way to capture those displacements are going to be correlated with the neural activity. And so our approach has been to use a technique called digital holography, in which we're transforming standard 2D images. We performed Fourier transfer to them, and then if you apply speckle reduction filters to them, you can extract phase information from the image. And you can form what's called a height map. And the reason why this is interesting and important is that the phase information that you've extracted is 100 times more sensitive than you have with amplitude information. And so we wanted to see whether this technique applied to neural activity would allow us to identify individual neurons. So this is what we've been working on since 2019. Phase 2 concluded earlier this year, and the initial findings are extremely promising. We've been testing these in vivo in a rodent model. We've been finding metrics that are very comparable with what we expect to have, as I mentioned in the requirements that we have in the invasive capabilities. So spatial resolutions that are under 1 millimeter cube, temporal resolutions under 5 milliseconds, high accuracy. This is through 4 millimeters of skull phantom, and they were stable for more than 4 hours, and a field of view of greater than 9 millimeters square. Recall that the Utah electrode arrays cover an area of approximately 4 millimeters square. These experiments are still ongoing. Testing is still ongoing in animals, in this in vivo rodent model. But we are in phase 3, and we're working on transitioning this to human. Where are we headed next? I have a picture here of Elon Musk, because Elon has been investing funds through Neuralink because he's worried about the technological singularity. We're not quite there. However, we do take the approach that we want to provide capabilities to our users that go beyond what they're currently capable of. In other words, if, as you can see, our participant, he suffers from a spinal cord injury at C5, C6 level, but he does have some residual capabilities. We want to make sure that by implanting our neural interfaces, we are giving him capabilities that he currently doesn't have. And so this was an example of something that we weren't sure could be done, in which he is interacting in this virtual environment with virtual objects. And when he comes in virtual contact with these virtual objects, each one of these cubes that you see here is associated with a different stimulation characteristic, and we're asking him to put them in order of how he's perceiving them to be from the weakest to the strongest.
That's the strongest. That's the weakest.
As you can see, did a phenomenal job with these 4 different intensities of the stimulation. Was that
4, 6, 8, 10.
that's the end of the talk. There's a lot of people to thank and who contributed to all the research over the course of these many years, collaborators, team members, funding sources. There's a lot. I have the good fortune of being able to work with all these extraordinary members and collaborators, friends. And so the last thing I wanted to do was make sure I gave a plug to the NER conference that's coming up, and which is going to be home turf for us. Make sure that you have you're thinking about it. Deadline just got extended to November 19th. So thank you.
Thank you very much. We really appreciate it, and good overview of what's been going on, and we're looking forward to it. NER in the spring as well. Are there any questions from the audience now that people want to jump in?
If I can just say first one thing, Fred, and others at APL that have joined, our old friends, Matt and Brock and Robert. And so thank you all for coming. And one thing I want to do is to bring this work more to the attention of our large army of masters and PhD students. So I'll work with Mark and others so that more people here know about it. We'll have to do better scheduling, I think. Okay, but great talk.
Sounds good. Yeah, this is what these are for, right? Helping to tie the bonds.
Yeah, well, so if I, the last one is, of course, it's amazing, the holographic, and as you said, the mechanism isn't kind of fully understood. But irrespective of that, I understood that that's a good temporal resolution, the way it works. How about the spatial resolution? I mean, is that going to be like a general hologram of the whole area, or is it like kind of pixelated for local areas where you can localize an activity?
Yeah, we're going to be. It's basically going to be an image, right? You see an image, and you can extract information from that image. We are keyed on the displacements that we're going to be able to perceive of these individual neurons, or close to that, right? Neural
yeah, but
activity
so that but it's a hologram,
resolution.
Right? But it's like a hologram. So does it have a literally the way we think about a column or a pixel of a neuron, say, yep, that's where that spot is active, or is it you get a kind of blurred whole region image?
I don't. We'll have to see exactly what it ends up looking like, but so far it's been at a pretty high spatial resolution, so you can see within a really small area the activity that's happening. Is it single units? I don't know. I think we're close enough that it's approaching multi-unit to maybe electrocorticography activity is the way I'm looking at it.
Great. Any other questions? All right. Well, thank you everyone again for joining and sticking around, and thank you to Prof for the talk. And hopefully we'll see you guys in the near term.
Hey,
Thanks everyone. Have a good day.